For today, we're going to be talking about control of microbial growth. There are many reasons why we would want to control the growth of microorganisms, primarily because we, of course, don't want them to grow in places and times where we don't necessarily want them. There are many different examples of antimicrobial techniques that have been used throughout many ages, and people typically performed these practices without really understanding what they were doing or how they worked. And you'll see that in these different examples here. A lot of them are very similar to some of the practices that we have today, but without the knowledge of how microorganisms carry disease and transferred it from person to person, it was almost like a roll of the dice that these things worked as well as they did. In ancient Egypt, for example, people used fire sterilization. In Greece, you could burn sulfur to fumigate buildings to get rid of pests. Hebrews would burn contaminated clothing, and smoke was even commonly used to kill fungal infections for many centuries. We now know that the use of things like heat and chemicals have very profound effects on the survivability of different bacterial species. And as I mentioned, these are things that we still have in practice to this very day. A lot of these methods could even be based in the contexts of culture or religion, or even in perceived functionality. People that were treating others that had the Black Plague would wear special masks that would apparently help to prevent infection of people in close proximity to those that were unwell. And the plague mask is one of the most iconic signs of plagues of the past. There are many reasons why we would want to be able to control microbial growth, but the two that affect human populations the most are disease and food spoilage. If we can control microbial growth, we're able to destroy pathogens and prevent their transmission, to prevent our food from spoiling or from people getting sick in the first place. And we can also reduce or eliminate microorganisms that are responsible for the contamination of things that we need in the day-to-day, like water, food, and other substances. Even though we have been able to develop a lot of techniques around this type of thing, they're still not always going to be readily accessible because some of them come at a very high cost. If you think of something like UV sterilization of water, if you're looking in places of the world that need that the most, in places where, for example, you have to take medication to go if you're traveling, just in case you might end up with some kind of pathogen that can cause intestinal distress, those places don't necessarily have the means or the infrastructure to implement some of these things. So while we do have basic knowledge of how to control different types of microorganisms, it's still not always accessible everywhere. So there's always R&D going into these topics to make them more accessible and attainable for all people. The three main methods that we use to control microbial growth include chemical agents, physical agents, and biological agents. We're going to look at a couple of examples of each of these today, and hopefully you'll get a good appreciation for all of the different things that you could even be doing in your own very day-to-day. This is a really great summary. We're not going to touch on all of these things today, but there are definitely many different topics in here that we will be kind of glancing over or at least addressing. One of the biggest differentials on how we approach decontamination and sterilization is what we're actually sterilizing. Are we sterilizing something that's alive, or are we sterilizing something that is non-living? In terms of inanimate objects, there is a lot of terminology that is associated with that versus things that are performed for living tissue, and being able to distinguish those is important. So let's run through some of these definitions. When we're talking about inanimate objects, when we look specifically at sterilization, this is the destruction or removal of all microorganisms. Disinfection is going to substantially reduce the number of a microbial population, but it may not necessarily get rid of everything. You're attempting to kill or inhibit most of the microbes, if not all, that may cause disease. There's also sanitization. This will also reduce the number of microbes to levels that are considered safe. So again, while it may not destroy all of them, it will at least reduce them in numbers to an amount that will make them safer. When we look at microbial control for living tissues, these are some terms. We have antisepsis. Antiseptics, you've probably heard of. Those are the chemical agents, of course, that will destroy or inhibit microbial growth and spread. There's also the process of degerming, where you're going to remove surface microbes when performing surgeries or an injection. If we've had vaccines or other types of needles, they'll take that little alcohol pad and clean up the skin before injecting you. That's a great example of degerming. Something like chemotherapy, will use chemical agents to kill or inhibit the growth of microorganisms in living tissues. So this is something that's typically applied systemically. So it will be added to somebody's bloodstream, allowed to travel freely throughout the body, and have a widespread effect. Perhaps not a specific effect, depending. If we look at chemotherapy, for example, that targets cancers, it's not necessarily only going to target cancer cells. It will target them, but among other cells, which is why we see so many people that undergo chemotherapy not feeling so great, because it is not as specific as other methods. It's also important to distinguish here some of the terminology and how they differ. So when we look at the suffixes or the end parts of words, if something ends in "cide," this is meant to kill. Bactericides, fungicides, algicides, viricides, those are all things that kill the things that they are meant to, kill their targets. If something is static, like bacteriostatic or fungistatic, it means that it will inhibit their growth but not necessarily kill them. When it gets removed, microorganisms can grow again. So you'll see these things applied in certain cases, depending on what the needs might be. We've talked about this a little bit in the past, but just to bring it back around here in a new context, the pattern of death in microbial populations is not instantaneous. You will never be able to kill every microorganism the first go because death by an agent is typically exponential. If you apply an agent to a surface that's contaminated, 90% of the population will be killed in each interval. And typically, a lot of things that we do apply in sterilization techniques do require a certain time frame of exposure. So it's not just spray and wipe necessarily, but things do need to sit for a little while to have their effects. There's also the possibility that in some populations, the final cells that are going to die might be resistant or somehow be able to subvert the effects of the agent that has been applied. And if that's the case, they may in fact take longer to die. There's also the potential for viable but non-culturable cells, or those VBNCs that we talked about a while back, that can also be a big problem. They usually require some kind of culture technique to check for death to make sure that cells have not just gone into some kind of dormancy state for a little while and can spring on back later. So when we try to measure the effectiveness of an antimicrobial agent, there are 6 different factors that we look at. It's the population size, the composition of the population, the concentration or the intensity of the antimicrobial agent, the contact time that we already touched on, the temperature, and also the local environment. Population size is kind of self-explanatory because, as we just mentioned, death is going to be exponential. An equal fraction of the population will be killed during each interval, and when we talk about intervals, we're more or less talking about a segment of time or a length of time. A larger population will take considerably longer to kill than one that's small. The larger the population is, the larger the 10% left over after each interval will be, which means that the larger your population is, the longer it's going to take to die off. This is a great little table just illustrating an example of this. And you can see in this last column here, the log 10 of survivors, it takes 6 whole minutes of that antimicrobial agent being applied to kill everything. If you were to, for example, apply it and remove it too soon, you could still have survivors, and those bacteria may start to divide again and eventually repopulate themselves. So this is a great chart just to kind of reiterate what we've talked about. If you have a big difference in the number of cells that are being treated with an antimicrobial agent, the larger the population, or the higher population load as it's shown here, the longer it's going to take to actually kill all of the cells within that group. You can see here, lower populations will die more quickly because of all the things that we just mentioned. The composition of populations also has a large effect. We know that there's a high potential for endospore formers to exist if they are from Bacillus or Clostridium genera. And obviously those are very difficult to kill compared to normal vegetative-only cells, but also the age of cells can have an effect. Younger cells are typically easier to kill. And this is mostly just because of a lack of experience. We did touch on last lecture the ability for different bacteria to share around antimicrobial genes, plasmid genes that can carry things on them that will allow bacteria to persist better. And not having had a chance to have those experiences and exchange some DNA through conjugation may put younger cells at risk. There are also a variety of different microorganisms that can withstand adverse conditions as well, and an example of these we've already discussed is Mycobacterium species. These, again, have that waxy cell wall that makes them impermeable to a lot of the things that we would normally treat them with as a physical characteristic of their anatomy. That's not to mention all of the other adaptive features that cells can have that are shown over here in this diagram, like efflux pumps. If a cell is treated with an antimicrobial, it may have developed a mechanism to kick that antimicrobial out of their cells so that it can't affect them. It may also have other types of mechanisms at the enzymatic level, where if an antimicrobial agent is detected, it gets either destroyed or altered so that it's not effective. Trying to treat different types of microorganisms is a very difficult thing, even if you know what you're dealing with, because you're constantly trying to dodge all of these different mechanisms and permutations to be able to kill bacteria effectively. The concentration or the intensity of an agent will also have a large effect on how effective it is. Usually, if you're using more of something, the quicker microorganisms will be killed, because as you increase its concentration, the better or more potent it will be. There are exceptions though, and you may notice that a lot of resources talk about using 70% ethanol. That's a lot more effective than using 90% when we disinfect surfaces, and this has been shown with different types of tests over the years, and that's why 70% ethanol is usually used in the laboratory setting when we were to spray down benches and make sure that they're tidy and clean before and after we do our work. Temperature also plays into this equation because at a higher temperature, you can use less antimicrobial agent to kill bacteria. This is probably due to that increase in molecular motion that we see with Brownian motion. Your chemical agent is going to reach its target more effectively at a higher temperature than it would at a lower one. Again, that duration of exposure is also important. We have to make sure that we're reading labels and making sure that we are applying our antimicrobial agents for the appropriate time frame. Taking them away too early can result in populations not being fully killed and having a chance to bounce back. Environmental conditions can also either help or hinder microbial destruction. Things like acidic substances are easier to pasteurize than things that are basic. And if you haven't heard of pasteurization, it's a sterilization method that we'll talk about later. You've probably heard about pasteurized milk or pasteurized honey, but having an acidic starting material helps to enhance that process. If there is the presence of higher organic matter, that can also help to protect microorganisms from destruction. Biofilms are made by bacterial communities, where a lot of them will lay down different kinds of sugars and other types of molecules like proteins and nucleic acids to make themselves a very thick and impenetrable cave-like structure on the surfaces of where they may live. We see these a lot in the human mouth, in the teeth, and every time you have plaque on your teeth and you're having that scraped off at your dental cleaning, that's what they're actually removing. That's a type of biofilm. Biofilms can also form on medical implants and a variety of other things in nature, of course, or in industry. So being able to manage these is pretty important. We'll talk now about some physical control methods, and we're going to touch on each of the ones that are mentioned here. We'll start out with heat. We can use dry heat, which would be fire or warm air. We have moist heat, which is water or steam, which is very effective and typically what we use quite a lot. It will kill a ton of different microorganisms, including yeasts, molds, bacteria, and viruses. And it will also degrade molecules that tend to be a little bit more difficult to break down, like nucleic acids or DNA. It can also help to denature proteins and disrupt cell membranes. You can also do boiling, but boiling will disinfect very well, but it does not necessarily sterilize. Again, remember, sterilizing means the removal of all microorganisms from whatever you're trying to cleanse. Endospores are very highly resistant to even hours of boiling. You can boil them forever. You put them into culture and they'll sprout right up. So certain types of microorganisms do require autoclaving. This is a great little table that kind of shows some of the differences between different organisms and how long they would actually need for heat killing, and specifically moist heat killing, so usually with water or with steam. For vegetative cells, those are ones that are reproducing, they're alive, they don't have an endospore, they may have a capsule, they all have their cell walls. They're just normal cells that are not spore formed. And then you can see in the spore column the other side of the coin. So when we look at yeasts and bacteria, you can see that those two are the easiest to kill at the temperature ranges that they have shown here, where molds and viruses tend to be a little bit more difficult to destroy. In this other column though, you can see some pretty normal numbers like 5 minutes, 30 minutes, 0.5 to 12 minutes. And then we have 2 to over 800 minutes at 100 degrees. 800 minutes is a long time to try to kill a single-celled organism, so these bacterial spore formers are extremely resistant to death. Autoclaving is a method that uses steam and pressure to sterilize. And this is what we actually use when we prepare media, plates, and anything else that we're using in the laboratory to manipulate. It will kill all vegetative cells and bacterial endospores. And what happens is you put everything into a chamber and it has a door that has a gasket on it that seals. Steam enters the chamber because you add some water into it, and as the temperature rises, it starts to boil. It produces that steam, which increases the pressure and the temperature of the internal space. And typically we heat it to 121 degrees Celsius and 15 psi for 15 to 20 minutes. After that depressurizing step, everything's going to be really hot for a little bit, but it's going to be sterilized. There shouldn't be anything left alive in those materials if autoclaving was successful. The process is also used to process biological waste. So anything that we would produce in the lab, like old plates or old cultures that we need to kill, have to go through autoclaving prior to being put into the regular waste. We certainly wouldn't want to just release a bunch of living microorganisms out into the world, so we have to make sure that they're killed before they go out. Autoclaving is the best method to ensure that things are being adequately killed. So this is an example of a large-scale industrial autoclave. The one that we used at the university is quite a bit smaller, but the same principles apply. So you have this door that seals, as I mentioned, which opens up into this large chamber. So you load everything in there, and then you add some water. You can see down here there's a whole plumbing system involved that will allow for water after the fact to get drained out. But you add water to it, you seal it up, and then you hit go. That temperature will get up to that 121 degrees, 15 psi. You leave it in there for a while, and then at the end of it all, everything is sterilized. Depending on how many objects and what kind of objects you're autoclaving, you may have to put it in for more or less time. Instruments and dry materials can go in for less, whereas if you're autoclaving liquids like agar or medium, that will have to stay in for a little bit longer. And for the record, you do not need to know the different components of this machine. This is just here for your information. Unfortunately, not everything can be autoclaved. Heat-sensitive objects cannot be subjected to this type of thing, so we have to think of alternative methods that do use heat, and pasteurization is one of them. To pasteurize something, we heat materials to 55 or 60 degrees for a couple of minutes, and that is enough to sufficiently kill non-spore-forming bacteria. If you're dealing with spore formers, autoclaving is the only option, but for those that do not form spores, this is perfectly fine. This has been applied to milk and other food products. We have high temperature short time and ultra high temperature pasteurization, with the differences in temperature and duration as listed here. And on the level of industry, when we are producing these types of foods in large amounts, the amount of equipment that is required is quite vast. So you can see that this is a typical factory where we would have a pasteurization site where materials could be heated and cooled and packaged up in a sterile environment to prevent that spoilage that we've been talking about. Now, you might be wondering about objects that can't be autoclaved but are contaminated by spore formers, and there is another option called tyndallization. This is an intermittent sterilization where you heat by steaming for 30 to 60 minutes, you incubate for 24 hours, and then you repeat that several times. That's going to allow for sufficient amounts of repetitive strain and stress that could probably end up killing some of these organisms. We regularly perform dry heat sterilization in the laboratory. Incineration by fire is something that we do all the time on the bench by putting our tools into the flame and allowing them to get red hot. This is an example over here of a bacterial incinerator, which achieves the same thing as a flame. I think I did mention it in the lab, but it does it in a way that keeps materials enclosed. So you're not burning off material into the air around you, but instead it can burn off in a closed off area so that you're not more at risk of inhaling or taking in those aerosols if they get produced. You can also put stuff into an oven. Putting things in the oven at 160 or 70 degrees Celsius for 2 to 3 hours is usually enough to destroy most microorganisms and, of course, denature cellular proteins. While it may work well, it is considerably less effective than most moist sterilization techniques. A great example of this is Clostridium botulinum, which can cause botulism. It can be killed with 5 minutes of autoclaving, but it takes up to 2 hours in an oven to die. And again, those are spore formers, so that's a specific example, but it's just to show that even though you have an effective method, there are sometimes better methods out there depending on what you're working with. And knowing what you're working with is always the first step. Talking about temperatures some more here, there's a variable that we like to determine called the thermal death time that's defined as the shortest time that's needed to kill all organisms in a microbial suspension at a specific temperature under defined conditions. This is known as the D value, which is the time required to kill 90% of an organism or a spore. And you might be wondering, well, why is that useful? Well, it allows us to be able to compare microorganisms to one another and plan effectively as scientists. If you know that you're going to be working with materials that do require a certain type of sterilization method or certain conditions in order to be able to sterilize them, you can prepare in advance. You can't really have a lab where you cannot kill things prior to throwing them into the landfill. So these types of considerations are very useful. Low temperature can also be very effective at killing microorganisms. Freezing at minus 20 degrees Celsius or lower will stop or slow metabolism. It will also kill bacteria as ice starts to form. Liquids are typically what microorganisms are suspended in, and as those freeze, they can form ice crystals that can penetrate cell walls and pierce cell membranes and essentially skewer the cells that are present in those foods. That's not to say that frozen food is completely devoid of microorganisms or sterilized, because it can very well contain microorganisms. You're supposed to slowly thaw frozen meat. A lot of people will throw it in the sink, throw it in some hot water, and thaw it under those conditions. Actually, you're supposed to be putting it in your fridge and giving it adequate time to warm up to fridge temperatures and thaw over maybe a day or two versus the same day. And this is just part of food safety, because if you do happen to thaw things in the sink, they have the potential to wake up on you and can infect you. High pressure is another method that we use, and you've probably eaten something out of a jar or a can in your lifetime. So pressure can help to alter the structure and integrity of cellular proteins and other kinds of carbohydrates, which can halt metabolism and stop bacteria from growing. The advantage of canning and jarring is that you're going to be able to preserve a lot of the awesomeness of your foods. You're not going to really have to alter your flavors or your colors or your nutrient values versus some of the other methods that we'll be talking about shortly. But you also have to consider that canning and jarring was something that was widely used even prior to refrigeration, because it is a very effective method at preventing microbial growth. Desiccation is another workaround where we can dehydrate different kinds of foods, because most bacteria and other types of microorganisms require water. If you pull all the water out of your food, essentially you're removing one of the main components needed for life, and dehydrated foods will last a very long time. As always, there are exceptions. Some bacteria can survive for a very long time without water, but most will die within 1 hour without it. Unfortunately, again, endospore formers are the exception, and they just go into their dormancy state and wait for a time when water comes back around. So again, knowing what you're working with is important. Osmotic pressure that's applied from differing the solute concentrations inside and outside of cells can also be an effective way at preventing them from growing. Adding high salt or sugar concentrations into foods like dried meats or even jams or jellies can cause any bacteria that are living within them to die due to osmotic stress. If you're creating an extremely hypertonic condition outside of those cells, now let's think about this. What happens to that water in those cells? It comes out, right? Cells crenate and they shrivel up and eventually die as a result. So messing with that osmotic balance is an excellent way to kill things. Once again, that's not to say that they cannot support life, but it will prevent most bacteria from being able to grow. It's also possible to filter bacteria out, and this can be used for heat-sensitive liquids or gases that are not easily applied to the other methods that we've discussed. There are two main types of filters. We have depth filters or membrane filters. Depth filters are going to contain fibrous or granular materials that are bound into a thick layer that's filled with channels of a small diameter. Typically porcelain or asbestos can be used, and this is a size-exclusive method that's going to prevent passage of very small things and filter them out of a solution. So the molecules of the liquid will be able to pass through just fine, but a big chunky microorganism may not be able to fit through. We also have membrane filters that have porous membranes with pores of a known size, so a similar type of application here. And these can be made from a variety of different materials that do have some size exclusivity. Membrane filters are predominantly the ones that are used the most because they do have that known pore size, and there are a variety of pore sizes to choose from. The filtration is also more uniform and predictable. So how this method is performed is usually by something called vacuum filtration, where you're going to pour your material into a little funnel that's going to have the membrane in it, and then you attach a vacuum hose to suck the liquid down through the filter quickly. And anything that is too large to pass through is going to get stuck on the filter, and everything that is meant to be sterilized will continue to pass on through. So you can see an example of the apparatus right here. So this is going to contain a little thin filter paper inside of it. This is the liquid that we're filtering. So that's going to get passed into the top through the filter. And then there's a vacuum mechanism applied here to suck that liquid down into the vessel. So all of this liquid has been filtered. All of this is unfiltered. This is also a great schematic, just showing that a little bit further. You can see the porous nature of the filter material, and all of these microorganisms are too big to fit through the pores. So they stay above, and all of that liquid that gets sucked through by vacuum filtration will be devoid of microorganisms as a result. Here is some great electron microscopy showing some more of that, where we've got our filter material on the bottom here. And you can see in both examples, we've got some bacillus cells that are stuck. They can't get through, of course. Over here, we've got some cocci. So a very effective method, but of course, and I'll say this about 1,000 more times, I'm sure, you need to know what you're working with because you need to know what pore size to use. We also use HEPA filtration. You've probably heard of things using HEPA filtration if you've got an air purifier in your home or even for furnace filters. And these high efficiency particulate air filters will remove 99.97% of particles that are less than 0.3 micrometers in size. These are used extensively in biological safety cabinets where we do a lot of bacterial prep. The airflow in those places is constantly being run through a HEPA filter, which provides a sterile area to work that does not have to necessarily be around a Bunsen burner flame. There's also the idea of N95 masks, which does something very similar to wearing a HEPA filter on your face because it will prevent the inhalation of very small particles of around the same size. We've seen these being used quite a lot in labs and hospitals and in the public forum where people are out and about. So this here shows somebody working at a biological safety cabinet. You can see that they're working inside of what's called the hood, essentially, and the airflow in here gets pulled in and comes up and around and gets filtered prior to coming back down into this main chamber. So there's a little bit of a vent here at the front where air gets sucked in. It comes on up through the machine and then gets blown back down onto the workspace, and it gets recycled that way. So it's constantly being filtered. So you can open up your agar plates and your cultures in there, and if everything is running accordingly, you should not have any contamination of your specimens. Radiation can also be used to sterilize, and there's two main types that we would use. There's ultraviolet or ionizing radiation. Ultraviolet is probably the safest for us, even though it's not all that safe. There are precautions that come with using any kind of radiation, of course. But the way that ultraviolet light acts is by preventing DNA replication and transcription, primarily through the introduction of thymine dimers. So it will actually take two adjacent thymines in DNA and connect them. When all of your replication machinery comes along in an attempt to actually move over that section of the DNA, it gets physically hindered. So it will prevent DNA replication or the transcription of any genes that those thymine dimers happen to be a part of. This is a very common technique when we look at sterilizing non-living things, especially things like water. And we'll be able to see some great examples of how UV radiation actually affects bacteria in the lab as well. Now we'll switch over to chemical agents. Because physical agents are most often used to sterilize objects, chemicals are usually used for disinfection or antisepsis and can be used on living and non-living things. There are risks that we run by implementing certain techniques because over time, bacteria are extremely adaptive and they can in fact adapt to a lot of the chemicals that we would normally apply. Resistance to chemicals is very prevalent and it's not as widespread as antibiotic resistance, but some microorganisms have actually gained the ability to pump chemicals out of the cells. So those efflux pumps that we discussed are things that we're seeing more and more and will prevent our methods from being as effective as we may want them to be. There has been a very large push, especially in the dental hygiene industry, to use mouthwash a lot. And what we're seeing these days is a very high resistance to mouthwash and the chemicals that are involved in those types of antiseptics. As a result, they're becoming less effective at the work that they're supposed to be doing. There's another group of chemicals called phenolics that can be used as surface disinfectants, and these were first used by Joseph Lister back in the late 1800s to reduce the risk of infection during surgery. We talked about how he was using a phenol mix before going and working on patients, and because of that observation, other doctors took that on as well and started to eliminate or at least limit the amount of disease that was being spread in hospitals. The advantages of phenolics are that they're long-acting and effective, and they're even effective against tuberculosis. Tuberculosis causes lung infections and is very difficult to treat, but phenolics are effective against them. The disadvantages are odors or skin irritations from prolonged use. Alcohols are probably the most common disinfectant and antiseptic that we have available to us. Primarily ethanol and isopropanol are the ones that we would see most often. They're effective at killing bacteria and fungi and function by denaturing proteins and also dissolving the lipids of the cell membranes. When we soak things in alcohols, that will effectively disinfect small instruments, but it can also be used in tinctures. And tinctures are usually combinations of different chemicals that are being used for disinfecting. So they're usually used in tandem with other things, sometimes alone, but also with others as well. So you can see in this diagram, some of the effectiveness of different types of disinfectants has been measured here. We have soap and water, all the way down to 1% iodine and 70% ethanol, which is a tincture of iodine, which is the most effective. We have time here on the x-axis, the percentage of bacteria that are surviving, and you can see that the steepest line down will be the most effective at killing bacteria the fastest and the best. Halogens are another group of chemicals that are very effective at disinfecting. We're probably the most familiar here with chlorine that we would use in things like bleach, or even in larger bodies of water like in pools or hot tubs, for example. But regardless of the application that you're most familiar with, all of the variants that use chlorine will produce hypochlorous acid, which will kill by oxidizing the cells. Iodine is a well-known skin disinfectant and will help to oxidize cellular components as well. The problem with iodine is it can cause skin reactions and other kinds of allergies. Chlorine is also bad if it's inhaled. And one good tip I have for you is if you're ever going to use bleach in your bathroom, make sure you do it with cold water because you don't want to make that go into the air in the steam of a hot room and inhale it. So always use bleach with cold water. That's a lesson you don't want to have to learn twice. Heavy metals and quats are also effective, but they're obviously dangerous to work with because they can also affect us. But things like mercury, silver, arsenic, zinc, and copper have been used as germicides for many, many years, and they have an extremely high toxicity. Unfortunately, they don't tend to be germicidal. They're only germostatic, so they can limit the growth of the organisms that they're being applied to, but once they're removed, growth can resume. An example of metals still being used in our very own day-to-day is 1% silver nitrate that gets added to newborn eyes to prevent ophthalmic gonorrhea. So while many of these methods are not safe for long-term use or for regular exposure, in the short term they can be very effective and helpful. There are also quats that can be used in detergents, and these, of course, like many other detergents and their additives, work by eliminating microbial membranes. They squeeze into those phospholipids, causing their dissociation. And in addition, they can also bind to other organic compounds. By doing so, they prevent them from being able to interact with others or eliminate their functionality by restricting their movement. We also use aldehydes. The most common one that might ring a bell is formaldehyde, which is a long-term preservative, and these work by inactivating nucleic acids as well as proteins. They're very effective disinfectants that can kill even spores, and that's why we preserve tissues in them for long-term storage. Sterilizing gases are also used, so these are chemicals that can be applied in high concentrations to eliminate microorganisms, and an example of this is ethylene oxide, which can be used for the sterilization of things like linens or catheters, syringes, petri dishes, especially in the healthcare setting. These work by blocking DNA replication and halting enzymatic function. In the advent of treating diseases, we learned about chemotherapeutic agents that can be applied and can effectively destroy microorganisms in living hosts. These are selectively toxic chemicals that can target specific groups of organisms, and they can be used internally to kill or inhibit microbial growth. I wanted to mention them here because they're obviously an important part of this conversation, but we're going to take a large part of another lecture to talk about them in more detail, so we'll leave it there for now. When we are attempting to evaluate the effectiveness of a disinfectant, there are a lot of different ways to be able to do this. We're going to look at this in the lab as well, but here are a couple of tests. We can use the dilution tests, where you would take different bacteria and you dry them onto rings, and then add them to different concentrations of disinfectant, and then culture them and see if they grow. And depending on the effectiveness of the antimicrobial agent, they may grow, they may not, they may grow more, they may grow less. You can ascertain a lot of that from that type of exposure. More commonly, we would use the disc diffusion method, where you have these little paper discs that are soaked in different disinfectants or have different antibiotics applied to them, and then you would apply them to a bacterial lawn. If you're wondering what that is, you may remember doing spread plates, where we have a uniform layer of bacteria that gets spread out. And if you apply the material via this paper disc to the surface, you'll notice that bacteria may not grow in their general vicinity, which would indicate that that would be an effective treatment against that certain type of organism. So depending on what type of organism you're growing, you may see different amounts of effectiveness depending on what you're applying. But within an experiment, you can actually measure what's called the zone of inhibition. And generally speaking, the larger the zone, the more effective the agent is. That's not always true, but for our purposes, we'll say that it is. So in this example here, you can see against Staphylococcus aureus, chlorine is the most effective, with hexachlorophene following behind, with then quat, and then O-phenylphenol coming up the rear. Interestingly, there's also the idea of biological control of microorganisms, where we pit one kind of microorganism against another. A great mechanism for this is through bacteriophages, because we know that they are able to hunt down and attack and infect bacteria. Bacteriophages, again, are a kind of virus. What I'd like you to take away from these images are A, B, and C, where they have applied a phage to bacteria, and A was the earliest time point, B was partway through their experiment. This was 1 minute following the application of the phage, and C here is after 15. So you can see that these cells in A are nice and happy, they're vegetative. After only a minute, a lot of them have been destroyed. And by the time we get over to C, a lot of them are dead. So this is an effective mechanism if you can make it work. Biological control involves a new field that will examine the predation of bacteria by other bacteria, or also virally, like we just discussed. So anytime you are using another living organism or an acellular non-living virus to kill another microorganism in a living host, this is the kind of stuff that we're talking about. Vibrio is another great example of an organism that can also do this. They will actually bore holes into Pseudomonas and effectively kill them. There are a couple of videos here that I'll share with you that you're welcome to click on and take a look at, but Vibrio is a very promising intervention, and so is phage therapy as well. So with all that said, I suppose I'll leave it here for now. Thank you so much for your attention today, and if you do have any questions, you know where to find me at laurar@nipissingU.ca. Enjoy the rest of your day and take care for now.